Good morning, everyone. My name is Justin Howard. I'm bringing my second presentation from biosystematics on DNA sequence data and DNA models of evolution. Hope to keep this concise. I'm going to cut out and really heavily edit all my ums and pauses to make this a little more brief for you and tolerable. This is a very interesting subject, and I don't think I've studied anything more in the last 2 years with my time at Western that it's more philosophical almost than just biological. There's a lot of complicated statistical theory and just ideas about how we should count things in this field, and it's just been very interesting, very challenging, and I've really enjoyed it. So what I want to talk about today is just 1 or 2 slides on how they faced challenges before this DNA information was available to them, and then the increasing challenge of organizing this data as it became available to us. There's been an explosion as everybody started to sequence DNA information and put it into different data banks. That posed some challenges in how we're supposed to analyze it, how we're supposed to retrieve it, and then we have to analyze it within the context of a model of evolution. And maybe I'll be able to explain a couple of those correctly and concisely for you. So it seems like everybody always starts out with a Darwin quote, but in this one it caught my eye because it seems like he was sort of lamenting the fact that he knew about evolutionary changes within organisms and ancestry, but he was never going to actually see it happen. He could only observe it as it is at that time. When he did publish his theory and everybody started to get on board with it, it really caused some issues in how we're classifying things. Ultimately, everybody was sort of split into 2 camps. Some were saying, well, if you continue to base classification of organisms or grouping them by morphological characteristics, that's subjective. It's based off of their opinion, whereas this new DNA sequence information is a little bit more independent or objective. It's repeatable, and so that should be the basis. We had to figure out what we're going to do with the fossil record, because that was a big part of dating some of these relationships and divergences or rise of new species. I believe the consensus today, the final, where we've ended up at is using all of that information and combining it. And this paper right here that Dr. Phillips provided to us, it's a chapter from an online book. The author was Brent Mischler. Really put all of that into context for me and helped me to really understand it. If we're going to do deep phylogenetic studies and also look at these more recent or fast-changing relationships when we're trying to discern a new medicine or where this disease is coming from, this bad phenotype, we're going to need to use all of that. And so one of the challenges that Darwin faced, just for an example, was here. I've got some bats, 2 winged animals here, and obviously he might have grouped those together. But when we go back and look at the molecular data, we understand that these bats, which are mammals, are more closely related to gorillas rather than birds. I would not put alligators more closely related to eagles than monitor lizards. The DNA sequence data doesn't lie. That's what it's one of the ways we're able to reorganize some of these trees that we've used to discern relationships between groups of organisms. So just to get a couple terms down before we proceed, one of the challenges that was faced before molecular data came about was the problem of homology. The bats and the eagle that I showed you in the previous slide, they would ultimately be grouped together, but they're not actually homologous traits. They are observable and they do look like each other. But when we group organisms today, we can either support or refute those groupings based on a phylogenetic analysis. So ultimately, like I said before, we're going to use both of these types of observations and data, whether it's from the fossil record or from morphological data, to group things. One more great example, this little graphic here I've got, you would obviously put these 2 animals with wings together, but in fact, parakeets or birds are more related to the T-Rex rather than the pterosaurs. It was easy for me to get a little sidetracked on looking at this sequence data and deciding which paradigm I'm going to be looking at it from. And by paradigm, I mean I can be looking at the nucleotide sequences and how they're directly related or homologous to another organism, or I can look at the RNA that's encoded by it, or the proteins, the functional proteins that have similar jobs between different groups of organisms. At each level or each paradigm here that I'm looking at, I can decide relationships, and some of them are more useful than others in certain contexts, and others are not. It really gets a little bit crazy right here when you're talking about amino acids, because more than one codon can code for a functional amino acid, and amino acids can be changed out, but a protein can still have a pretty similar function. And so things can get lost within that translation there, and it can become a little bit tricky when you're trying to classify things. It's important to remember when you're viewing all this data from different angles, whether it be from a codon or a protein or a DNA sequence, to remember this principle of parsimony here. In an ideal situation and any good systematics, as Mischler stated, you would put your data into a matrix first before you started to build a tree. And technically, you would want that tree to be reversed or be reversible to a matrix, and you would have the same data present. That's a repeatable experiment. He called out the idea that the data matrix is largely ignored during your efforts on building a tree, and that systemists will ultimately focus on all these different methods of analyzing a tree, whether it's Bayesian inference, maximum likelihood, or neighbor joining, rather than discerning if they had their data placed correctly into a matrix. And that's where the crux of this DNA sequence and models of evolution comes into. Maybe I can articulate why this is. Some of the pros of using molecular data, specifically DNA data, in phylogenetic studies would be being able to rather quickly discern genetic causes of a trait that I'm interested in. But say I have a group of organisms that's hardy and withstanding low temperatures or desiccation, like a group of bacteria or fungus or some kind of insect. I can compare closely related or similar sister taxa and discern where that lies within code or within a gene. I can understand critical points of divergence in looking at ancient relationships. I can take a known regulatory part of a gene that we have sequence data for and compare it to a wide group of taxa and discern if they have the same thing or not. That would be useful if I discovered a new organism and I had a sequence of its DNA data. I could compare it quickly to a large swath of taxa and decide where it should fit on the tree. David Mischler stated in the paper I showed you earlier that ultimately scrutinizing DNA sequences is the workhorse for a systemist trying to classify things, and it underpins every other way we may compare data, like for example, whether it's an RNA transcript or a functional protein or an area in a gene that produces the protein. It all is going to rely on that DNA molecular data. Some of the downsides would be that, consider that I have a gene located in one area of a chromosome for one organism, and I want to compare that to another organism. Well, that gene or that sequence could be located in a completely different place on the chromosome, and so that presents a problem when comparing sequences. You may miss it when you're doing a blast search. DNA has a tendency to evolve in a pretty consistent manner, and so if I'm going to go back and look at an ancient relationship, I would want that change to be frozen and not change every time I added some new data to my phylogenetic tree during, bootstrapping or reconfiguration from a new sequence that I may need to add. Sequences that are similar or homologous, they can be conserved widely, but what if it's a analogous trait or homoplasy? If I decided that these two organisms are related because of their similar sequence data, that could be error. That could be wrong because there could be an issue of convergent evolution going on there. And finally, when from a statistical standpoint, DNA has 4 nucleotides that can ultimately be compared. And when you start adding in other things like RNA or protein sequences, you've got a lot more things to compare, which allows for a wide range in probability statistics. So DNA is limited in that sense by only having 4 character states. So let's look at some of those pitfalls and hopefully understand why we have nucleotide substitution models and ultimately why we weigh our data. take this first example here. Between these two sequences that I'm comparing, I'm going to be able to see that pretty easily. But if you have this example here, say we've had one change and then another change, well this change within the sequence right here is going to be lost. And I'm only going to see right here, I'm only going to see these two sequences when I ultimately compare them. And I'll miss out on that data. Down here is a good example of convergence. I believe I made this up, or this diagram. I had an initial change here and then it changed back. So now I think that these two relationships, or these two sequences, are homologous, but originally there was a new species that arrived here. I can further this idea of a problem you might encounter when trying to figure out lineages is with this diagram, on which I added mine too. So if this is convergence, I've just added it right here onto this slide and borrowing this diagram. And so these dark lines indicate the main lineages, and these smaller letters, these uncapitalized letters here, will be species that are extant, they're alive, and then these diamond ones indicate species that are extinct. And so if I had a split here, like I've indicated in my diagram, and this species became extinct, but I'm looking at it here, I would assume that A is more closely related to C rather than this divergent species that is not around anymore for me to appreciate in my sequence data. Here's another issue you can run into. Again, we have the main lineages indicated in green, and inside would be the clade we're going to end up with at the end of our diagram, the tips of our phylogenetic tree, and inside these little ones indicate smaller lineages that may not have lasted. But according to A over here, B diverged from A a long time ago, versus over here it's a lot more recent, this ancestor. And so because of issues like this, we need different models of evolution to provide different algorithms in our analysis, in our computer analysis of all this sequence data we're coming up with. This is one type of phylogenetic tree I'm not too familiar with. It's called a network tree. But I do know that under Bayesian inference and the Markov chain Monte Carlo model, the MCMC, which you might have seen different options for that when you're looking through Mega and other software. This is why that has to happen, or this is why that is there to increase our probability and allow us to give better statistical results when we're analyzing things. So to do this, we have to make sure we're weighting our data initially, and just like I stated earlier, a good systemist is making sure his matrix is correct and that this data is good to go before they're trying to build a tree and then compare that tree to other trees or add data to it and evaluate it. So one of the simplest, or probably the simplest model, would be the Jukes-Cantor. It states that nucleotide frequencies are weighted equally. Mutation rates are is the different parameter that we're looking up at. And I simply made these right here, these diagrams or this table, it just helps to illustrate the mathematical concept behind it. I don't want to teach you something about math here because I could not do that myself, honestly. But if I take my sequence 1 and sequence 2 and I compare them, did a multiple sequence alignment, and I've made the homologous sites line up. If I were to give a score to each of these matches here, remember this is all nucleotide frequencies are weighted equally, and so out of 4 nucleotides you can have them each show up roughly 0.25% of the time. But when there's a match, ultimately if you were to go across and add this up, all these rows would equal 1. On the other hand, the transition rates, all these rows would equal 0. Maybe that will give you a little bit of an idea about how these algorithms are working and how they're weighting individual characters, which would be individual nucleotides. Once this is done initially, it's going to take that result and then compare it to another sequence. And if you have multiple sequences in alignment, it would do it over and over again, just sort of compiling all that data and giving you a final score to look at and building your initial phylogenetic tree that you will use to compare with other hypotheses. Here is the next substitution model that I want to tell you about. This actually should just say it should calculate transversions as an additional parameter because the transition rate stays the same as it did prior to in the Jukes-Cantor. Transversions happen when a purine will exchanged for pyrimidine. This happens less often than a transition where a purine will be exchanged for a purine. And so they give it a special score. And through the wizardry and magic of calculus, we'll come up with this formula that somehow is a ratio, eventually produce a gamma distribution with a different type of score that will make up your initial phylogenetic tree when you're doing these comparisons. And so that tree will be compared to others. It will be available for you to test your hypothesis about other phylogenetic relationships. Overall, multiple sequence alignments are the starting point for these analyses. They're solely for the point of taking a homologous sequence and comparing those sequences and lining them up with homologous nucleotide bases that they contain. And those columns will eventually be indicative, or you'll be able to eventually tell where their selective pressures, and they can inform you of different relationships and different or changes in functions or phenotypes. So there's many other different models out there, and they give different weights to different parameters or add other parameters to that algorithm. And really, just like we were shown with that handout that Dr. Phillips gave us, when you add more than 2 taxa, basically, the results become exponential very quickly. And so does the computing strength needed to compare different sequences, especially if they're much longer with many nucleotides in them. It really will just depend on, do you want a fast computation, or do you have time to wait and give you a better educated guess? Here's one example of a multiple sequence alignment that I did with my phylogeny of interest. This was part of a pairwise comparison where they took 37 different taxa and compared it pairwise for each one. So this block, you would have 37 of them in your results. And down here we get these things called a consensus. Once I've done my comparison between my alignment and just maybe, let's just say, a blast search, I may end up choosing that result to go search for other unknown sequences, or to build my initial phylogenetic tree here from this data, and then that can be compared to a reference sequence. This is my reference sequence for this comparison here. So depending on the length of your sequences, how many different types of sequences you have, will help you to choose which type of alignment algorithm you want to scrutinize your data with. Distance-based methods will take an alignment between the two you have, or yours and a reference sequence, and convert that into a matrix with a score and produce a single tree. And from there, that tree will be compared to other sequences and other trees that may or may not already have data attached to them, like fossil records. Some examples of them are the UPGMA and the neighbor joining method. They both have their advantages and disadvantages in terms of speed, computing power required. Some of these, like distance-based, once you lock that first tree in, if you want to go back and compare different sequences and add other data to it later, whatever you have initially in there is going to remain in there. Whereas more so in the character-based methods, it will consider those changes. Examples of these are maximum parsimony, maximum likelihood, and different types of Bayesian analysis. So I would dare to say, I may not be completely correct on it, that these types of comparisons or multiple sequence alignments are more for a quick analysis to find out just where you're at on the map, or maybe to find a quick similarity or homologue between your unknown sequence, for example, and what's out there. And this is more for a deep analysis, looking at deep ancestral state type comparisons. That concludes my presentation. I thank you all for sticking through with me, and I wish everybody the best of luck through the end of your class. Have a great rest of your week.